Hello, everybody. Especially in this context, when we suffer the consequences of the pandemic disease, right now we have the responsibility to take these studies and these analyses in a new approach, an evolutive approach. The Thymic Stromal Lymphopoyetin, this special cytokine, have the responsibility to promote the inflammatory response with the mastocytes and even create a feedback with the type 2 immunity response. This last cytokine promote allergic diseases. Overproduction of this cytokine exists in many syndromes with an inflammatory approach and allergic approach too. Exists a diversity of cells that can secrete or receive the signal of TSLP and produce this kind of receptors. This diversity of cells can be sensory neurons, that it's so important because the olfactory neurons are the target of the COVID-19 disease, and it is a great damage, especially in this area, with smelling loss, like a symptom in the COVID-19 disease. However, we can see that this pattern of TSLP has suffered processes linked to the natural selection and competition too, becoming a selective pressure response to pathogens environmental as individual level or a population level. But how this signal has a leading role in these pathologies and how this has a relation with the SARS-CoV-2 infection and how this is a response of natural selection, competition, and evolution of cause. As a natural selection, we can see that the airway pollution depress the immune system. That's the selective pressure. The last research to fight against these pathogens with a depressed immune system is a strong inflammatory pattern that can replace or reduce the interaction of some factors of recovery. Recently, studies linked the diversity of inflammatory response of these TSLP signals into isoforms, the large isoform and a short isoform. With this kind of diversity of the TSLP sequence, we can see that exists a competition, one more time, between types of the TSLP in the organism. And what's the solution of the organism to select these tools to fight against the diseases or infection? The first form is the typical signal with a tetramer receptor, the TSLPR receptor, and the interleucin alpha-1 receptor. These kind receptors create a complex to receive the signal of the TSLP. And the TSLP excision due to this tetramer receptor can promote an inflammatory pattern from moderate to severe in many infections or allergical patterns, such as the atopic asthma. In contrast, in the short isoform, exist some properties that are improved, such as regenerative signal and antibacterial, anti-inflammatory effects, and even antiviral effects. This discovery can explain how this selective pressure is induced in a positive selection of the large isoform due to the lack of recognition skills, recovery of tissue properties, and of course, the result of the suppression of innate and memory cells action against new pathogens or common infections in the respiratory tract. Although the allergical percentage in human populations is considered minimum, less than 1%, this pattern of immunity suppression for the environmental changes in the airway composition are a main factor to promote the susceptibility to the allergical and severe diseases with the critical inflammatory syndromes. We can see that the incidence of allergic asthma is a consequence of the life quality and genotype variants in the populations. In Japan and China, for example, we can see a high level of asthma patients in comparison of other industrialized countries around the global north. The effect in the asthma development and COVID-19 patients are unknown because of the randoms. However, Shank and other collaborators So that the COVID-19 patients in the 0.01% of asthmatic population had severe problems in a 0.005% of the people. This case can be related with the kind of strain due to the constant pattern TSLP large-isoform related in the atopic asthma. So what is the competition in this study? The TSLP polymorphism's prevalence is the key in a population level, because we can see in this analysis of the Mr. Harada that all of these polymorphisms, especially one of the three polymorphisms, is the origin of the atopic asthma allergic disease, especially in this population of Japan, is because the prevalence of this phenotype around the population. The angiotensin receptor enzyme 2 and the serine protease in the T cells layer are the mechanism to invade the organism, the host organism in the human beings for this virus. These kind of receptors and enzymes complex create a K-log complex between the spike protein in the layer of the coronavirus and the receptors that like ACE2 or even the serine protease complex in the T cells layer. The virus promotes an autoimmune response with pro-inflammatory cytokines and lymphocytes T helpers, regulators and B cells to promote a strong cytokines. Complexes such as interferons alpha and beta are considered an antiviral effect to reduce the ACE2 receptor levels, and the interferons and serine protease presence are linked with the TSLP secretion too, as antagonist in the case of interferon and agonist factor in the case of the serine protease. So that means that the TSLP secretion is related with the stimulation of serine protease secretion. And without the serine protease secretion, the TSLP is degraded. And in the same time, the interferons production, especially with the interferons type 1, like interferon alpha and interferon beta, are related with the absence of the TSLP. In a study of the moderate and severe COVID-19 diseases, researchers Lucas Carolina et al. In 2020 could see that the severe and high mortality of patients have a high levels of TSLP in the incubation period. That means between the 15 days or 20 days. And in the last part of the pneumonia, the TSLP are decreased as a consequence, of course, of the toll receptors, lymphocytes, and the dendritic cells destruction because of the cytokine storms and factors with TSLP crossed stimulation and inflammatory factors of the tumoral necrosis factor alpha, interleucin 17, interleucin 5, and interferon gamma are presented in the high levels in flux cytometry in plasma and lung fluids. In contrast, the moderate disease has a constant and low frequency of typical TSLP large form. That is the main reason, because this form of the TSLP have presence in this kind of severe diseases with more frequency. However, some regenerative and growth factors and antibacterial, anti-inflammatory, and antiviral signals were promoted in the moderate and normal disease. Can believe that the short variation can act in that 75% of population that have a moderate disease. In vaccination, for example, exist some aspects in the eosinophilia was so common in the vaccination process for the allergic people that are neutralized by two ways. The anti-TSLP or antibodies against the TSLP secretion, that the presence of the TSLP, especially the large form. So the prevalence of this short variation is the key as positive selection in the novel disease to promote a better response. And this TSLP factor can link with the existence of a therapeutic site in a severe diseases of COVID-19. Thank you so much.